From the JAMA Network, this is JAMA Ophthalmology Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Ophthalmology.
Hello and welcome to this author interview from the JAMA Network. I'm Neil Bressler, Editor-in-Chief of JAMA Ophthalmology, and today we're going to look at biomarker detection and validation for corneal involvement in patients who have acute infectious conjunctivitis. And joining me is the lead author of that article, Dr. Gerami Seitzman. Dr. Seitzman is a professor of ophthalmology at the University of California in San Francisco. Gerami, thank you for joining us.
Thank you for the invitation.
Maybe we can just start for the non-cornea specialists, such as myself. When you have acute infectious conjunctivitis, can you remind me what we're talking about in terms of what corneal involvement is with this conjunctivitis?
Absolutely. So when we're talking about acute infectious conjunctivitis, I think a lot of people will refer to this as pink eye. There are several different pathogens that can cause acute infectious conjunctivitis, and many times it's just the conjunctivitis. But sometimes this will turn into a keratoconjunctivitis where the cornea becomes involved. And so when the cornea becomes involved in the setting of infectious conjunctivitis, we become a little more concerned with that clinical presentation.
And so you wanted to see if you could identify certain biomarkers that say, this acute infectious conjunctivitis is going to get corneal involvement, or that it already has corneal involvement?
Well, in a perfect world, a biomarker would predict who would develop an outcome. So we were curious if we could identify a biomarker that could accurately predict the population that would develop corneal involvement in the setting of infectious conjunctivitis.
And what made you want to do this with your investigators in the first place? Why might it be important to discover some biomarkers in someone with acute infectious conjunctivitis? Why would that be important to know?
Well, there are a lot of reasons why this is the case. First of all, this is a really fun study to talk about because it is a really multidisciplinary collaborative project. So it's a group of us internationally that get to work together to try to answer some really interesting questions. We have a lot of deep sequence analysis that's being looked at in this project, and all of the sequencing is coming out of my colleague Thuy Duan's laboratory. She's an incredible uveitis specialist that runs a world-class ophthalmic genetics laboratory. We have a bunch of bioinformatics analyses, and that's being led by my colleague, Dr. Ermin Hinderwerth. But this study is taking place within the context of our NIH-sponsored study called Scorpio, which is this really cool collaborative effort of 23 sites over 16 countries worldwide, where together we're studying the etiologies of infectious conjunctivitis over the entire globe. So we're working as a group together to try to answer some interesting questions. And one of those questions are, can we identify, of this huge population of infectious conjunctivitis, can we identify the smaller subset of people who would subsequently develop or have developed corneal involvement so that we can identify a population that perhaps would warrant closer investigation, perhaps different treatment as well?
That makes a lot of sense. And can you give me a little idea of what are some biomarkers one might look for? What were you thinking about investigating? Not necessarily all the ones that you found or discovered, but what were some ideas that you had at the start as to what may be a good biomarker?
Well, I think sort of a general question is, what is a biomarker in general? And that's something that we can measure quantitatively that can help predict a certain disease state. And with RNA deep sequence analysis, which is the tool we're using in this NIH-sponsored study Scorpio, we get so much information about all the genes that are being transcribed at once during an infection. And so rather than saying, I wonder if this particular gene were going to be expressed, we have this great opportunity to really ask an unbiased hypothesis to say, of all of these tens and tens of thousands of genes that are expressed, can we identify a signal that would lead us to look for a biomarker? In other words, can we use our data to help us identify the biomarker? So we use sort of an unbiased hypothesis to do a comparative sequence analysis, which led us to a set of genes that we could further interrogate. And that led us to finding one particular gene that looked like it was giving us a signal where it would be a good biomarker.
And before we get into what you actually discovered, how much of this was planned at the time that you got this group together around the world to begin to investigate these cases of conjunctivitis? Were these pre-specified ideas, or was this something that came out of perhaps some other investigations that you already had started?
When we started Scorpio, which is this international conjunctivitis study, our primary aim was to determine the etiologies of infectious conjunctivitis worldwide. And just a quick word about that, because we find it a little interesting. In the United States, I think when people hear pink eye, we're like, what a nuisance.
Mm-hmm.
If we had to clean down the lane, we got to close down this little area, or maybe it went through our family. If you're the patient, it's certainly a nuisance if your wife gives it to you or your kid gives it to you. Sometimes infectious conjunctivitis goes through a school or a nursery school or a daycare. But what we don't think about a lot is that in other parts of the world, specifically Asia and Africa, infectious conjunctivitis happens regularly, often seasonally, and in numbers that are hard to even conceive of, where tens of thousands, sometimes hundreds of thousands of people are experiencing massive epidemics of infectious conjunctivitis that really shut down school systems and regions of a country. And curiously, those etiologies often go uninvestigated. So the primary goal was to just determine what are the pathogens responsible for infectious conjunctivitis. And then as secondary goals, we were interested in exploring some of the host response transcriptome information, meaning if we weren't able to identify a pathogen, could we guess by perhaps a signature that our host immune response was giving us what pathogen it was by our response? So at the beginning of the study, we sort of knew we wanted to capture the Dwan Lab's ability to do great RNA sequence analysis to look for pathogens and also look for host immune responses. And then as a subset of this, for several years, we were wondering, well, gosh, we're starting to explore these comparative sequence analysis to look for differentially transcribed genes. And that led to the question is, with the differential expression analysis, could we identify something that could possibly be a biomarker in this setting?
So that really gives it to us in context. And how many cases of conjunctivitis were you actually able to explore this question in?
This study takes place in 3 stages. The first stage, we used what we call convenient samples. So samples that the Dwan Lab already had that we had collected years back, a population of about 60 infectious conjunctivitis swabs that were a combination of United States samples and samples from India. We collaborate with the Aravind Eye Institute in Madurai. So the first stage of this was to look for differential gene expression in that convenient sample. So we had the samples already in the lab. We knew there was a subset of patients that had corneal involvement and didn't have corneal involvement. And the Dwan Lab ran deep RNA sequence analysis on this population, and we did a comparative analysis to decide which are the genes that were most differentially expressed between those 2 populations. And that led us to one particular gene that sort of highlighted itself within this analysis.
So that was your detection. Then what's the validation? How do you determine, was this really valid, or did you just find one by perhaps chance alone?
Great. So what happened is that we compared a series of different classifying algorithms to determine which predictive statistic algorithm was best able to determine if our population had corneal involvement or non-involvement. And that turned out to be just good old-fashioned logistic regression. So we used our logistic regression analysis and we compared the performance of how well logistic regression could predict corneal involvement with these 13 genes that we identified as most differentially expressed. And then we did something fun where we repeated the analysis, removing one gene. So a take one out analysis. We had 13 genes that were differentially expressed. We removed one gene and we saw how well did the logistic regression perform. And it turns out when we removed one particular gene, namely it turned out to be the ApoE gene, the performance of the logistic regression dropped significantly. significantly. So we thought, oh, well, that's interesting. Maybe this ApoE gene is contributing really significantly to the performance of the model. So that was then turned into part 2 of the study, where we said, well, let's check that out. We have the population. Let's take the patients where there's no corneal involvement and is corneal involvement. And the Dwan lab ran RT-PCR to amplify ApoE in that population. And sure enough, ApoE was expressed at much higher levels in the population that had corneal involvement. So we thought, maybe there is something here. And then the third part of the study is where we said, let's harness the power of Scorpio. Let's look at populations that haven't been looked at yet, populations that weren't used to train the classifying algorithms. So we have an additional over 100 patients from India and about 60 patients from Thailand. And we looked prospectively at the ApoE expression in those populations to see how well did a certain threshold of ApoE expression predict corneal involvement. And that was the exciting third part, where at a certain threshold that we identified as balancing ideal sensitivity and specificity of the assay, we could predict that ApoE could predict corneal involvement with a pretty decent specificity of about 88% in the India involvement and 93% in the group from Thailand.
So that is pretty exciting to be able to do this. I guess the next question is, should this be part of our clinical practice, or is it worth being part of our clinical practice? Can you just follow all people, and see who gets corneal involvement? Or will this really help narrow down the ones that perhaps need greater monitoring?
Well, right now, this is exploratory, and we think, this is an interesting finding. But the greater picture of it, it gives this exciting idea. When we think about people with pink eye, the vast majority of those patients do not present to ophthalmology and certainly don't present to a corneal specialist. We only get them when there's complications or something really affecting their cornea that would diminish their visual acuity. But when you think about a population at large with infectious conjunctivitis, most people are bothered. Most people have some vision complaint, photophobia, decreased vision, and they're being screened elsewhere. So in a perfect world, if, for example, we had the ability to test a biomarker. So just imagine for a moment that someone with infectious conjunctivitis goes to urgent care and someone had the ability to swab them and say, you test positive for a biomarker that would predict corneal involvement. Then all of a sudden, out of this big population with infectious conjunctivitis, we may have been able to screen a smaller population where we say, it actually is worth the resources to send you for more subspecialty evaluation because we think you may develop corneal involvement on the basis of this, because obviously most people don't have the ability to do a slit lamp examination in a screening primary care environment.
And what are the limitations so far? Does this require independent confirmation, or do we have enough information to say we're ready to run with this?
I don't think we have enough information to say we're ready to run with it yet. Certainly, I think we need a few more larger sample prospective studies where we take it out for a spin and we say, let's try to identify this biomarker in this population and really confirm how well that works.
And are you planning any of these next steps? What are the next steps to be done, or are others planning to investigate this? What have you heard, or what are you thinking about?
Well, this is a surprise when we got to ApoE, first of all, because if a protein's allowed to be famous, ApoE isn't famous in ophthalmology at all. We don't really think about that protein. It has some relationship with Alzheimer's disease. The specific variant ApoE4 is associated with the development of Alzheimer's disease. But in the world of cornea, we almost never talk about it. It's been reported rarely with Schneider's corneal dystrophy, which is not entirely unsurprising because it has to do with a lipid deposition. But we first need to do a little investigation of whether or not this makes clinical sense in the setting of infectious conjunctivitis. So I'm really looking forward to exploring what ApoE can mean in the context of the cornea. And also another different analysis that we could think about is here we're just analyzing the performance of one protein, but in the future we're really also interested in evaluating if certain combinations of proteins, so different, maybe more than one together, would increase not only the specificity but the sensitivity of the assay. So I think we're really looking forward to refining the analysis, looking to see how other proteins may or may not contribute to the predictive capability of the model. And then also importantly, testing this in many different populations across the globe outside of Asia.
Well, I really thank you so much for explaining the beginning of this investigation and the exciting findings you had, and really what some of the next steps may be to help address this problem that's really going on worldwide. And it would be great to be able to narrow down our resources and perhaps identify people that may be at greater risk and need greater follow-up or more specialized follow-up. So thank you so much for sharing this with us.
Thanks for the conversation.
We've been speaking with Dr. Gerami Seitzman, and she's been discussing the work that she and her colleagues have been doing in the Scorpio investigation, looking at biomarker detection and validation for corneal involvement in patients with acute infectious conjunctivitis. I'm Neil Bressler, and you can find a link to this article in this episode's description. And to follow this and other network podcasts, please visit us online at jamanetworkaudio.com or search for JAMA Network wherever you get your podcasts. This episode was produced by Daniel Musisi at the JAMA Network, and thanks for listening.